Today I'm delighted to introduce one of my postdocs, Tony, who's been here, time flies, since March 2019, doing a lot of interesting work related to brain studies and fMRI. He's also very proud of many of his accomplishments, including some tools he's developed, Easy fMRI and Easy Data. Today he's going to talk about one of our joint projects about shared space transfer learning, analyzing multi-site fMRI data. And Tony's around, so talk to him after the talk today, or he's very accessible. Just give him a shot. I'm sure he'd be glad to talk about what you're doing and be able to figure out how to help out with that. Tony, all yours.
Hello, everyone. I'm Tony. I'm postdoc working originally with Professor Ross Greiner and Professor Andrew Greenshaw, respectively, from Computer Science Department and Psychiatrics Department. So today, this is actually my second AI seminar. And by contrast of like the previous one, today I'm just talking only for one paper, which published in NUAA 2020. The title is Shared Space Transfer Learning, and I'm gonna talk about different concepts related to our paper. So this is the outline of my talk today. First, I just introduce what is task-based fMRI, and then I'm talking about some concepts coming originally from neuroscience community called functional alignment. So last decade, a lot of neuroscientists and computer scientists extend the idea for single-site functional alignment. And today I want to talk how we can extend this one for multi-site fMRI analysis. What are the challenges, or how we can address this challenge? And we call our algorithm during the talk SSTL. Let's start to see what is the task-based fMRI analysis. So this is one fMRI machine. Patient just lay in the machine and we can collect the neural activity. We have originally two types of neuroimaging. One just show the anatomical of the brain, like the tissue, like gray matter, white matter, or CSF. And at the same time, we have functional neuroimaging. Functional neuroimaging show the neural activity, and fMRI is one of the best ones because it can provide for us a spatial and temporal resolution. And it's high dimensional. We have several challenges to just collect the data, but like last decade studies show that it can provide for us a very reliable way to collect functional activity when subjects doing the task. So what the means of task and how we can collect the data. So just imagine a single subject. We have fMRI scanner, the subject just lay in the scanner, and for instance, in my example, the subject try to just watch the visual stimuli. So there are gray scales of photos here, face, bottle, and chair, and we start to collect the neural activity when the subject just watching these visual stimuli. So we have some ROI in the subject brain. For instance, for the visual stimuli, we are interested in VT cortex or temporal cortex. VT cortex is a low level, visual stimuli effect on the brain, and temporal is the high level concept. So most of the time we just watch the temporal cortex for the high concepts like face, objects, other things. And we capture this neural activity. At the end, when the device just captures these things, we have some matrix of neural activity. And during my presentation, I called this matrix algebraic representation. The reason we call this one algebraic, and it's like matrix form, it's always like time points and voxels, because we have another way to present the neural activity called geometric representation. So we use the algebraic or matrix form of the presentation of like data for advanced like computational analysis. But we are using also the geometric representation for understanding how our theoretical like computational models works. So we switch between these two concepts during this presentation, but they are actually the same things. For instance, here we have 3 examples. And we, for each like example, we have one single time point. And for instance, in the first time point, the subject watched the gray scale of face. The second time point, the gray scale of shoe and chair. So we have 3, and for each of these neural activity, we have voxels here. Voxels is the 3D shape of pixel. It's the same concept. So it's a cube and we made our like brain neural activity based on these cubes. So this matrix here is the algebraic or matrix form of the presentation. But at the same time, just imagine we select 2 of these voxels. So we can just draw some coordinates-based or geometric representation of this neural activity like this one. So for instance, based on just these 2 dimensions, the face is located here, the shoe is here, and the chair is here. So here for clarity, first, we don't have just 2 voxels, it's thousands of voxels most of the time. These geometric representation help us to understand some concepts. During my presentation for the computational model, we are using this notation, XDS. D here represent this data set or the site, and S is the subject. So for instance, for site 2, subject 5, we have X25. And here our X has the form time points times voxels. So here we have 3 time points and 10 voxels. So it'll be like 3 by 10. So our data set here, since we have multi-site fMRI data set, we have 1 to capital D, and each data set has SD subjects. And we have for each site, we have TD time points, which can be different across different sites. And for all of them, we have V voxels. The reason we have V and we don't have VD is that we are interested to just analyze, for instance, whole brain data or a specific ROI, which can be the same across the sites. So it is clear, for instance, if the first column for the first site is like voxel 111 in 3D space, it cannot be like voxel 222 in the second site. It should be the same position in the brain. So that's the reason we extend our idea mostly for the whole brain data. And we have some standard space to register all of the images to the same size. So achieving the V here is easy and practical. And we also have this notation again. We have TDV for each subject data in a specific site. So we called whole of these things, in neuroscience, they call it representational space. In machine learning, we call this one vector space, but they're the same concepts at this, in different like literature. So this is fMRI, and we have tasks for this one. It's a little different with rest mode fMRI, where subject just lay, relax, and we get like the brain activity when the subject doing nothing in the fMRI. And here our goal is understanding how brain neural activity and cognitive stage will be changed based on these tasks. So any questions so far from here? All right. Right now, I want to just talk about some neuroscience concept called functional alignment. I did my PhD on this topic, and it's really interesting both in computational perspective and neuroscience perspective. So until now, I just talk about the one subject, Mr. Smith. But right now, we have a multi-subject fMRI data. It's clear if I just collect my brain activity and try to model that brain activity, it's not generalized because of my brain connectivity, environment, a lot of other things, genes and so on. So we prefer collect multi-subject data. And here we have the same fMRI device, single one here for a single site. And we have multiple subjects. We have subject 1, subject D. And we are running the same category of stimuli. So again, it's same category, not exactly the same photos. So they are watching the category of seizures, you can just see here, and the category of faces, which are here. So faces are different, but they are still faces, and the seizures for each subject is different. We try to diverse this image and collect more like diverse neural activity to make more generalized neural activity. But I mentioned something, the connectomy. It means that the environment and the genes will affect our brain. So it's obvious my brain and yours doesn't work exactly like each other, right? And that's why when we are collecting the neural activity and we just use the geometrical representation to just show how this neural activity can be shown in the vector space, we can just see that, for instance, here the face are located here in my brain. For instance, yours is here, and the seizures are here and here. And for fixing this difference, the function alignment theory just say that they are exactly the same, but they are noisy rotation of the same template. So this idea presented in 2011, it applied to different cognitive state, different stimuli, and we figure out probably this idea works very well for at least for brain decoding. So the idea here is that we make the rotation between subjects to just achieve something called common features space. And these common features are the noisy rotation of the original subject space. And we have some transformation, which is mathematically exactly rotation. And these transformation map all of the neural activity to shared space. And if, for instance, we want to just predict these things using like classification analysis, then we apply our classifier here instead of here. So it's very easy to just, for instance, discriminate this area by using the V1, right? The voxel 1, versus like just discriminating the space by using this one, right? And another part is that when we just like map these neural activities to the shared space, we can easily just apply the similarity analysis or like clustering, other kind of things, which Rust doesn't like it. So that's the main idea. And I would like to present this slide, which is from Hagsby, the person who the first time present this idea. In this YouTube link, you can just like watch this later. There are different researchers like the famous Tom Mitchell, Jack Gallant from UC Berkeley, and Hagsby, who present the idea, and other like computer scientists and neuroscientists. You know, the original experience they just run was like, they have two subjects here. They watch the Indiana Jones movie, and they trace some voxel value for the subject 1 and subject 2. And here we have different values of the same voxel during the time. So each dot here is the time points, and the same color means that they are the same time points. So the reds are for instance, the first time point, the orange are the seconds, and so on. So as you can see, by tracing these two voxels, when these subjects just watching the Indiana Jones movie, you can just see that there is some patterns. If you just rotate one of these subjects based on the origin, you can just achieve to the second one. So that's the main idea. That was that spot, like these guys just say that, all right, probably this idea works in neuroscience. The original paper published in Cell, but it doesn't have any like kind of formulation. After that, computer scientists try to formulate this one, and they figure out the idea here is actually equal to this objective function, which is actually CCA, canonical correlation analysis. So here we can introduce our single site functional alignment, and then we later just come back to this objective function to use it for extending the idea for multi-site fMRI analysis. So here we have a single site data set, including S1 to SD subjects, neural activity, and we try to maximize this neural activity, the correlation between this neural activity by using a rotation matrix, or in our case is transformation, because we put the rotation in on the shared space. And this common or shared space here is our aligned neural activity. So we use a transformation to map each neural activity to a shared space. And we would like here to make some orthonormal neural activity. So you know that each matrix transpose each matrix, if it will be like equal identity, that one is orthonormal and is equivalent to the rotation in mass. So we would like to have this rotation in the end, and we would like to produce a more independent and orthonormal vectors for each row of this matrix. So here again, D present the site which is equal D1 to D capital D, but here whole of the objective function is written for a single site. So we don't care about the whole of this equation, but we'll use it later for multi-site one. So we have SD subjects for this one. All of these X has TD time points and V voxels. So our assumption here is that the number of time points in single site is the same. It means that the subjects will see the same set of stimuli. For instance, they always watch 2 photos of faces, 3 photos of bottles, 4 photos of like chairs. But the number of these photos are same for the single site. It's easy because most of the time they are the same neuroscientist person who designed this experiment in single hospital. So that's what they are doing for like last decade. But here, and you know, since we have like XD is TD times V, we reduce the dimension of voxels and we just map them to K components, and our shared space just defined in the K component. So here we naturally also have the feature reduction. So this feature reduction can help us to reduce the sparsity of the data and get more effective features. All right, so that's the single site functional alignment. Any questions so far from here?
So just to confirm, in this slide, there's a superscript D, but we should ignore that for now.
Yeah, for now, we just use a small d, and that the reason we define it like this because we'll extend it in the multi-site one.
Gotcha. Okay, so I'll make sure. Thank you.
Yeah. All right, so let's talk about how we can optimize this objective function. I don't want to just really go through the optimization, but you know, I want to just present to you the main idea. For optimizing the proposed objective function, we first generate something called projection matrix or the hat matrix. And here we add some regularization term to be sure that for any shape of number of voxel and time points, our objective function still can be coverage and can be like achieved to something. So we define this one, and we assume the mapping is this combination. And based on these two assumptions, if we substitute these two things in our proposed objective function, we can remove the R here and then achieve to something like this. And the reason we put symbol here is that this objective function here is not exactly equivalent to this one. So the difference is this regularization term. You know, the second author of this paper, who has 2 PhDs in math, and I believe that he's here today, he's not talking with me for a year because I put this regularization term here. And yes, this is not exactly the same answer, but this is our best chance to just get the best answer. So, all right, that's probably is not mathematically completely equal, but this one guarantee for any size of V, we have this inverse here. Actually, the things we did in our paper, I don't want to really go for like that much like the details, but the thing is that we like put the SVD implementation of the, like we decompose X based on the SVD, substitute it in the projection matrix, and we end up to something where all of these like X matrix here will be diagonal and will be like the sigma. It's like U sigma V transpose in the SVD decomposition, and we end up to something diagonal. And just calculating the diagonal matrix for this inverse is way easier than a full matrix like rectangle matrix, because most of the time our voxel numbers is way bigger than like the time point. So I'm not going for the practical part, but we extend also the idea for like the parallel processing. Each subject can be processed parallelly, and then G will be sum of these like iterative based answer. So that's the very short like introduction for the optimization. I'll be happy after end of my presentation go for the details. So any question for the optimization part here or not?
Can you just remind us just the size of the problem? How many voxels are we talking about?
You know, it really depends to the problem. If we are going for ROI, we have most of the time around 10,000 time points, but it's at least 17,000 like voxels. And it can be just increased up to 2 million voxels, which most of neuroscientists want that one because we have really good resolution. And like each X here is 10,000 times 2 million. So it's, you know, our data set is at least like 300 GB or something. So we are talking about a really big problem. And again, based on learning theory, when we are talking about 10,000 samples versus 2 million voxels, we don't have enough samples to satisfy that number of voxels. So here we try to reduce the like size of problem by selecting components, by like putting the SVD, reducing the problem to like diagonal inverse or something. So it is a really big problem actually. It's not big data in the like the sense of machine learning, but it's high dimensional data, definitely. So any questions so far? All right, let's talk about what is the multi-site fMRI analysis. I would like first just say that all of the story right now you just listened was about the site A, hospital in Edmonton, in, I don't know, I'm coming from Iran, in Tehran or somewhere, right? And all of the things we have done was based on a single fMRI machine. But we don't want to just create some brain cognitive model that works only based on this machine, right? We want to extend it for other site. For instance, we want to learn from Edmonton data, just apply it to Calgary. That's the reason we are just doing the multi-site fMRI data analysis. Here, my example is for 2-site, but our model here can be extended for multi-site, learning from tons of sites and then again applying to other tons of sites. So we don't have any limitation for this one. So how we can extend this idea and why we have to extend this idea? So the first reason is that fMRI data is high dimensional. So it just returns to like Ross' question. So we have high dimensional and noisy data. It making sense you generate a shared data and then transfer this shared data across sites rather than just transferring all of these high dimensional multi-subject data one by one through the sites. So it makes the problem simple. Occam Razor just say that simple model works most of the time better. Another problem for like generalizing the cognitive models in the sense of machine learning and neuroscience is that fMRI scanners are expensive. Collecting data is expensive. So I didn't find any person just give me fMRI machine for birthday to me. So if you want to do that, I'll be happy. But the problem is that most of the data we are collecting is just like 5, 10, maximum 100 subjects. So by 100 subjects, you cannot really just generalize a cognitive model for vision or higher aspect, like watching movies or something. That's the reason, like collecting different size data and then combining them, making sense. And these days, increasing the number of public data set make this chance for us to apply multi-site fMRI data set and generalize our cognitive models. That's the reason we want to just use the same idea. And you guys probably just tell me, okay, Tony, we just add this like different subject from different size, concatenate them, and then apply the functional alignment. But it doesn't work actually. The first reason is something called temporal alignment. I mentioned during each site, we have the same set of categories of stimuli and same size of time points. It's easy when you have the same set of experiment design during the scan. For instance, when they watch like 2 photos of faces and 3 photos of like chairs. But between sites, most of the time, it's so, always neuroscientists collect data, then try to find a machine learning guy to analyze it. And it's so late to just redesign the experience, right? And that's why most of the time different like sites have different temporal alignment or number of time points. For instance, in Edmonton, subjects watch 2 photos of faces, but in Calgary, they watch 3 photos of faces. It's really controversial how we want to just align these things. We want to remove the third photo or we want to keep it somehow. So our paper can address this kind of problem. Another problem, which Ross is famous in the world for that one, is the batch effects. So simply, we can just say that environment can change our imaging quality. Different fMRI machines, one is Siemens, another one is General Electrics, they have different standards to save the images. So somehow we have to just deal with this kind of problem. So it's not really easy task to combine 2 data sets, like concatenating to like a matrix and then get really effective like classifier analysis based on that one. So that these reasons motivate us for making something called shared space transfer learning. So we are using transfer learning, obviously, for multi-site fMRI analysis. It's not the traditional concept of transfer learning generally in machine learning is a little different. Our SSTL can align homogeneous multi-site fMRI. So the only things you need is be sure that, for instance, you are aligning visual stimuli related to face, object, or other things, to visual stimuli to face, object, other things. It's not logically possible. We align the visual stimuli to audio cortex, right? This one is like wrong. The voxel value is wrong for this kind of things. The task is different. So the only condition we have here is that we try to align the homogeneous multi-site data set. Sorry. So another thing is that our method is not limited to overlap data set. So there are some multi-site fMRI data set using the same functional alignment idea, but they need, for instance, Tony just do a scan in Edmonton and Calgary at the same time. So based on neural activity generated by Tony's brain, they align these sites. So we don't need this kind of subjects. And again, if we are doing this one, it means that your model accuracy is based on that subject's brain, which is not guaranteed to be generalized across all brains in the world. All right. So we'll show that our alignment technique can improve the prediction rate across the sites. So let's start how the main idea works. So until now, we say that we have a site, for instance, D=1, site 1, and we have the fMRI collection for that one, and we show that how generate the common space. Let's do all of these things for other sites the same, exactly the same. So until here, the size data doesn't need to be combined really. So there are different subjects, different temporal alignment, probably different type of fMRI machine, but the task here is homogeneous. So we have same set of homogeneous cognitive tasks. And we are doing this one for all sides, and this procedure is completely unsupervised. So we just have the X, we don't have any label or other things. The idea here is that after generating this common space, for a set of training, we can map all of these common space to something called global shared space W. So this is the main idea. We map the shared spaces to some global shared space where is shared across all of the sites for the training set. And then after we generate this W, we can use it for the testing set as well. So how we can do this one? So this is a simple procedure. We concatenate all shared spaces across all training sites. Then we learn W as follows. It's like a KLT transformation, which is actually fancy like term for an extension of like the PCA, right? And we learn this one and we try to maximize the correlation between all of these shared spaces based on the W. So here we learn this objective function and then use W to map the training, testing data set. Here, I want to also like mention this one. We mentioned this one in our paper as well. So here we used a linear transformation. Yes, we can use autoencoders to achieve more nonlinear approach and more effective way. But the data set we had was really limited to 2-3 size. So this linear one works for us. But for real like practical approaches for real sites, we can actually use nonlinear transformation for which can produce better like result in comparison with our simple transformation here. All right, so I want to just like show you the different steps and a little about the algorithm. I try to just make this one very short to be sure that it will not be boring. So again, we have just 2 steps for our STL. The first step here is generating the common space G for each site independently, and then concatenate these G's in a single G here, and then map them to a global shared space. The algorithm should be something like this. And we have training set, which is multi-site data set. We have testing set, which is multi-site data set again, and we have some regularization term and number of components. This is that controversial regularization term for generating common space. And we have some common part, which is generating G, or the common space for all sites. This is across all training sites and testing site. Each site must generate this one independently. It means that if you are like the data analysis just mapping these things, each hospital can do this step by themselves and then send this G to you for applying the multi-site analysis. And this G here is TD, which is the number of components for a single subject, times K, which is the number of components, which like the totally, for instance, if you have 300 GB data for a single site, this one should be something like 100 MB, and that 100 megabyte is very easy to transfer by email or Google Drive than that 300 GB data set itself. So after that, we have the training phase, which is originally learning the W. We concatenate all common spaces, apply our KDT transformation or the PCA one, and then generate the W. And the things we are learning for like the classifier for all training sites is XDS, which is the neural activity of the subject S inside D, which is part of the training data set. R, which is map this X to the common space of that specific site, and the W, which map whole of these things to the global shared space, which is shared and common across all of the sites. And we learned this one as a classifier. And then for prediction, we apply the same procedure on the testing set. Here, the W will not be updated based on the testing data. The R is generated based on each site neural activities, all subject of the site, and the XDS is the subject S inside D for the testing set. And we applied this set of like mapping to our classifier to get the prediction rate, including like accuracy or like precision or any kind of standard like metric. So that's a whole of the procedure. Any questions so far from here?
Yeah, can I ask a question? So if you have like 5 sites and 10 people per site, you assume that the training includes people from all 5 sites, right?
No, we assume like we have completely independent training and testing, right?
Yeah.
For instance, we learn from 4 of these sites and apply to 1 of them, right?
Okay.
Or we learn from 3 of them, apply to 2 of them. You will see this one, Ross, in our experience design, and you see, for instance, we have site A, B, C, D, and apply and learn from each of them.
Just the notation makes it look like you have the same sites.
No, the notation actually doesn't look like, because here is from 1 to D hat, here is the D tilde, and here D tilde and D hat is the training.
That font doesn't come across. Okay.
Yeah, and that's why I just showed this one, try to just say, like,
Well,
all right. So,
and you're going to see what the task is. The task is for Mr. Smith. Is he looking at a chair right now?
yeah, and for instance, if it'll be like a visual stimuli, then it will be like watching grayscale photos. Let's see the data set right now. So empirical study. So our method is just compared with 6 different methods. First, the raw neural response in MNI. MNI is that space which we have a standard size of a brain for all subjects, without doing any TL or transfer learning methods. So here, we want to see that without doing any functional alignment, any other kind of like transfer learning approaches, how really the neural response works. And this is our baseline. And we compare our method with SRM, MDDL, and MDMS. These are the transfer learning method needs per site subject. So we need some subject be shared between sites for this kind, applying this kind of method. We also have 2 other like transfer learning algorithm. They are like ours, they are generalized transfer learning, but they are not multi-view actually. They are not using the idea of functional alignment. So we have these 2 also for doing the transfer learning. And we compare our method with these 6. So data sets. All of these data sets are public data set from Open Neural like website. So you can just see that, for instance, these 3 are from the same like data set. They have the shared subject. They are for a stopping signal with different like situation. So a stopping signal means that there is a key in the, and when we capture your brain activity in during the fMRI scan, you can press that key to stop based on some like kind of stimuli they give you. For instance, one of the most famous of them are like the gambling on balloon. There is a balloon here. Each time you can decide, press a button and increase the size of this balloon. And each time you press the button, you can get $10. But if the balloon explodes, then you lose all of the money. If you give up, then you can get that money you achieved that time and then leave the experience. And this kind of like decision making, how this kind of cognitive process works in your brain is the goal of, for instance, that task. Here we have the same things. This is the number of subjects. So we have 20 subjects for these 3 and 4 tasks, which is like pressing button, down pressing button, making decision and give up or just continue or something like that. And the number of time points, you can just see that the number of time points per data set is different. These are the data set who has like the same common subject with the data set. Like for instance, for A, we have the common subject with B and C, and like the same things for the others. And we also have that 4th data set like this 008, which is the same task, but doesn't have the same set of subjects here. And but they are related to A, B, C. So the cognitive task is decision making. We have Seaman and Flanker, which are the really famous tasks in cognitive science, and we can predict, they are almost a similar task somehow, and we can use them to predict the other ones. So they are just producing in different hospitals in the United States, so they are actually multi-site one. And we have also the last two dataset, there are different level of sweetness. So you like drink different sweetness level of water. So it's like water with 0 calorie, 100 calorie, 50 calorie, or so on. And we have some common subject between them, but we have different time points. And this is like our flavor study, our flavor data set. So these are the data set, and these are the set, and we indicate like data set by using A, B, C, and so on. So this is the first classification, multi-site classification analysis with common subject. So here we have some shared subject between sites. This symbol here means that in the first level of analysis, we learn from site A, and then apply on site B. And then we learn from site B, apply to the site A. And this one is the average of this classification analysis. So we report the detail in our supplementary material if someone wants to just see the numerical result. But here is the average of this one. So A to B, B to A to C, C to A, comparing B to C and G to H. So this is the flavor one. These are the established signal. And you can just see that the rate of classification analysis. And here again, we learn from site A, B, apply to C. We learn from site C, apply to A. So the average of this one is a little bigger than that one, so in some of them. And the reason is that we have more terrain set, at least in one site, right? And that's why we have better like alignment and better improvement. So this is for those data sets with shared subjects. And we also have classification analysis for those independent sites, which doesn't have any like common shared subject. So we learn, for instance, from A, B, C, and applied on D, and we, again, like learn from D, apply to each of these sites. And similarly, like for A and F, you can see that a little like that one was really like for specific for other not multi-view based like data set. These are like significantly reduced the classification analysis. And we have the same thing here. You can just see that here is a little better than the top one. The reason is that we have more data set here to satisfy our model. So, and again, here we have 2x2 comparison. So we report all of the numerical result. It's really hard to trace this thing, but the last bar is ours. And most of the time we like get better result by combining the functional alignment and the multi-site idea here. So here I want to just give you some more practical example. So for the dataset E and F, which is about the sweetness, which is the flanker and Simon task, we have two categories of like stimuli. For each category, we first generate the common space. So you can just see that these, for the first category, this is the average of neural activity in the common space. For the second category, this is the average of category voxel values across like this category. So this is the common space, and these are the global shared space. You can just see that these are exactly almost the same, not exactly the same. This is really like here, if you see that, for instance, here, this one is a little different, and there are tons of difference. You know, it's really hard by tracing colors, see the voxel values, but they are different, but they are way closer than like the independent site value. So this is how our like mappings are effective. So this is like visual similar. Ross always just called this kind of things beautiful, just beautiful, useless figures. So we have this kind of things to satisfy neuroscientists, not computer scientists. All right. So that's like the example. Another thing we mentioned, our method is designed based on the parallel processing GPU-based analysis, and that's the time point. You can just see that it's really like effective even in the runtime. It's like this one. You know, for instance, SRM, if it takes 10 seconds, ours takes like 1 second. And it's really like important, because most of the time learning from like high dimensional data takes long time, and having like this much of like efficiency for the runtime makes a lot of like good improvement of the analysis. So that's the runtime example for like these 4 data set, and the others has the same tendency. The reason is that we remove the iteration-based learning. We have a single iteration over a subject. So for learning the common space, we just visit each subject neural response once. But for instance, the SRM visited like, if we have 10 iterations, visit each subject 10 times. So that's the reason we have this much like difference between like the runtime. So that's it. Any question about the results so far? So as you may know, I just graduated from Nanjing University in China. We have a really good tradition. If like presenter asks, do you have any question? No one answering anything. Then presenter allowed to ask question from like listener. So if you want, we can just continue this like tradition here. But if we can just see the future works at the in the first place. So first, SSTL is a multi-site, multi-view fMRI analysis method. So we show that like our SSTL achieve better like result in comparison with other transfer learning result. The paper published originally like in December last year. And until now, we have received tons of like good feedbacks from other people. There are people in Turkey, United States, and China working on SSTL for theirs like multi-site dataset. And we get really good feedbacks from like the performance analysis and other parts. Our plan is using SSTL for the movie and data sets first. That one is a little complex in comparison with single task data set, and also other kind of like multi-site fMRI cognitive tasks. So this is a whole of like the summarization, but this is not all of the presentation. The first thing is that I want to mention the fMRI. This is actually the project I started from beginning of my PhD. Right now it has full pipeline of analysis, including pre-processing, feature analysis, model analysis, and visualization. It has all of my implementation of all of my papers, including the SSTL. And it also has tons of other like effective papers for task-based fMRI analysis. So you can see here, for instance, you can directly write Python code in the fMRI. It has some like data viewer for opening math file and something called EasyX. I'll talk after this one. And there are some like examples. This is like full cortex like visualization. This is kind of like similarity analysis generated by fMRI, and this is kind of dendrogram for different like visual stimuli. Here we just compare the similarity of these like neural response in the brain. So it's open source project. It's available in GitHub and GitLab. Welcome to just contribute, and we always like extend the toolbox for our new methods. So if you want to see our new like methods before even the publication, you probably need to like trace our code in fMRI. So that's it. But during the fMRI development, we end up something called EasyX, which is a library to save huge file in a single like file. So it is based on HDF5, but by contrast of HDF5, you can just save any non-homogeneous structure like dictionary, libraries, and other kind of like objects, anything. And you can save more than, for instance, in my case, I even save 150 GB data set in a single file. It's very easy to handle single file in comparison with tons of like files in the folder. So this one is we developed this one originally for Easy fMRI project, but we always welcome to if you want to use it in your other projects. It can exactly like save a dictionary in Python and just reiterative it again. So the main contribution of us in here in like the EasyX project is that you don't need tons of memory for saving this one. This library dump the memory from directly to a file, and again just can like return everything exactly the same format. It's by contrast of like MATLAB format, that one need some kind of coding. You cannot save more than 4 GB or something. So this is the second project. I also want to mention if you are interested in multi-site neuroimaging analysis, we have a research topic in Frontiers Neuroscience. The deadline for this research topic is for the abstract is end of August. For the manuscript is the end of like October. If you are interested, you have around like 20 days to just submit the abstract, and then for publication you have almost 2 months. And so here we are interested in same style of like analysis and like the research. So I put the link here. If you want, you can just go and just see the details. You can also see our other related study. We have NeurIPS AI paper in functional alignment in 2017. We have supervised hyperalignment in TCDS like journal. We have 2 other papers for reconstructing brain image, which is same, also almost same concept based on fMRI. And we recently extend our like research for practical application like pediatric anxiety, and it's published in scientific report. It will be like accessible in the scientific report website August 18. So there are the other related studies. You can download these slides using this QR code, and this is the direct link for our paper. And this is this one going to the Easy fMRI website directly. Thank you so much for your attention. And if you have any question, I'll be happy to answer.
Thank you very much. Great talk. I have a few questions, but let me give other people a chance to ask. And if not, well, let me start. So this really interesting idea of sort of you just rotate the brain around this high dimensional space. Why is that true?
I believe that I can I would like to just introduce this talk from Hagsby, who just like the first time answer this question. So the main idea here is that we are removing the effect of connectomy from the brain and achieve shared information across brains. And this is the science. So in the science, in like the scientific approach, we generate some model and try to just see how much our practical experience just show that this model works, right? And this is how they work actually. They just show in theirs experience, this is a noisy rotation of voxels. And this paper published in 2011, after that, there are tons of other
I'm not saying it's not true. I'm saying what does it mean to be true?
Yeah, I'm just going for that one, Ross. Be a little patient. All right, and after that, tons of other practical applications show that this one works. But why it's true? So let's talk about the mathematicians. So I believe that if Alessandro is still here, so he'll be happy for this one. So let's imagine here we are talking about the multi-view learning. So each subject neural activity is like a single view, and then for each of these views, we can just map these things to a shared space. just imagine the general PCA, right? That one just for a single view, try to maximize the correlation between the features, right? This is the same idea, but the thing is here is that we maximize the correlation somehow. All of the subject has this maximum correlation in the shared space. So this idea is kind of extension of PCA. So if the PCA works based on the idea, we maximize the variance of each feature or covariance between features, right? And then we have more chance to learn better model. Here also we are doing the same things, but for multi-view one, not for the single view. Like we don't have a single data. We have multiple subjects data. So this is the first things. And in the sense of rotation, why we are using rotation? So in math, we have different kind of like transformations, like scaling, sketching, or other kind of things, right? Rotation guarantee we are not changing the value, like we are not sizing the changing the size of vectors in the vector space, or like we are not scaling them, we are not removing parts of them, we just rotate them through the origin. So here we preserve the information. The only thing is that we change the representation of the information. So this is really important in like it's really agnostic actually. So that's why the rotation here is probably the most simple like transformation for this guy.
Let me just, I know Cloud will ask a question, but just wrap up. Imagine I had the same task being done, an R code on a Mac and a Python code on a Windows box. Same task. Are you claiming I could just do a rotation to transform the internal structure of the computers?
You know, if you are doing exactly the same task in the like.
I didn't say exactly, you know, comparable. I'm trying to recognize the fight scenes in Indiana Jones.
Yeah, but the thing is that Ross, brain doesn't work like a computer, right?
Oh no, I'm just saying, what does it say? I should let Cloud talk, but the question is just what does it say about human brains that it does this work, and not about general intelligence systems?
So based on my understanding, the things neuroscientists say that they are the collection of the neurons. And if the usage of oxygen in the brain doesn't have the same pattern, it means that there is some effect of like connectomies here. So they want to remove that effect and just get some shared like connection. So just imagine the social network. You probably have really crowded like connection. But if you get one of those like community detection like network for music or something, probably the connection between the like the music things are almost the same relation with the reality, right? For instance, although we have noisy connection of the subjects, if we remove those things, for instance, probably you have more a strong like connection between pop and hip hop than pop and rock, right? Or pop or jazz or something. So we are looking for that kind of like really abstract connection here actually. And we can say that, all right, we can never understand that noisy things in each subject, but this shared information probably is the same. And in the level we are, so we should never like forget, each voxel of like fMRI is around 10,000 neurons, right? So if we know that, you know, we have really a strong background, for instance, eyes are connected to the VT cortex. So we know that one is the place the visual stimuli will be processed in the different levels and then move to the temporal. We can cut the brain. I'm not recommending this one during my presentation, but we can cut the brain and see the neurons there. So we have evidence. And then, but the thing is that we are sure, we cannot be sure that in really like neuron level, they're just acting the same thing, but in the really big picture of the neurons, like 10,000 per voxel, they should be the same. So that's the neuroscientist like.
Thank you very much. Cloud's been waiting very patiently. Thank you. Great answer.
No, it's actually, well, you know, it's the same thing that I was asking, and I'm going to ask, you know, like, but from a slightly different angles, because I was asking Tony before, like when we actually worked together on this, or like 2 years ago, maybe when he first arrived, you know, like I was asking why does it work, right? So, and
you know, I want to just again just like answer this question in really like a general scientific approach. For instance, when Newton just say this is the gravity, we know this is the gravity working on the Earth, but not for really huge or really smart like things, right? And then we run a lot of experience, understood what is the boundaries of that one. Then Einstein extended for the huge things, or like the string theory or quantum mechanics just extended for the small things. We still don't know how really, for instance, the quantum physics does work, right? But we know that this is the fact. All of the experiments show that this one works. And for the brain, right now, we have the same things. Even, well, I believe that major of our knowledge about the science is about the space and like the physics than the brain, right?
Well, Tony, what you said actually was exactly my critiques about modern physics. But let's actually leave that alone. So my question, let me actually be more specific, actually, from a neuroscience angle, right? So do you think this only works in the scale of the fMRI, or it can even work for the single neuron, like smaller networks that, you
don't believe
like
that,
this rotation thing?
I don't believe that this one probably is exactly the same things in neuron level, right? Because probably we are in this like resolution, we will see like the straight line. But when we focus on the neurons, it will be a curve, big curve. You just see that one, like a line, right?
Yeah, I'm really curious, if it, so, like we have animal studies ongoing, but I'm really curious, like, on the individual neuron level, or at least in the causal imaging, right, in the local spaces, whether it's working, and then if it's not working or it's working, when you start.
I will note this one. The idea here we are working is not like a new idea in machine learning. It's a multi-view learning. We use it for analyzing audio, video, like normal audio, video, or other kind of like text mining, other things. We have a submitted paper in NeurIPS right now, and that one is extension of something published for the document mining. So we know that like increasing the correlation between different views, it making sense and makes like the analysis better. But yes, we have to be careful to use it, right?
Well, no, I'm not criticizing it. I'm actually thinking, like if it's for example, if it doesn't work on the neuronal level, then when they start to break down, will actually give us more idea about what's the principle of neuroscience.
So for those who doesn't know, we are working on the mice brain, task-based mice brain. This is like a collaboration between Claude, me, and Alan Chen. And we cut the mice brain, put some camera, and trace the neuron level neural activity. And we try to do the similar things, right? And I believe that probably that one is not the pixel level in that literature, but probably it will be some graph, and then we need to align those graphs. The idea of alignment is the same, but the way we are mathematically formulate them is different. So we still need to align those things again, but probably not exactly linear way like the things we are doing here.
Yeah, so basically, yeah, this is just a general comment. I'm really interested in, like from the principle of neuroscience perspective, whether we are actually capturing some very basic principles in neuroscience, or it's actually something that only works when it's grouped in a certain level, when you average certain neuronal activities, or maybe even worse, maybe it's only working on the blood response, blood oxygen response to some neuronal activities, but not actually the necessary real neuronal activities.
Our study right now is based on this assumption that fMRI has the linear relation with the neuron activity, and it makes sense because like we are tracing usage of oxygen, right?
Yeah, well, that's actually the one I mentioned was actually the part that most exciting for me.
Neuron doesn't like to use oxygen, just shows the wrong place in the brain somehow. Or when they are working, they like to share the oxygen with their neighbor, right?
Yeah, but anyways, I think this is like something worth to think. Like maybe we enter into the field of philosophy eventually, but I think that's something interesting. Yeah.
Yeah, but our practical applications show that this one at least works in fMRI literature, and there are tons of extension of the function alignment idea, since 2012 actually.
Mm-hmm. All right. Yeah, Russ had to head out. So yeah, great talk. Thank you, Tony.
Right. Thank you so much for coming.
Thank you, everyone. Thank you, Tony. And we'll see everybody in a couple weeks or so. Have a good weekend, everyone. Bye.
Thank you.